question, multiple choice. Know the difference between specific and non-specific immune responses, and like the first, second, third line of defense, like that stuff. No, like inflammatory response, we talked about that. Know about MHC class 1s, MHC class 2s, we talked about that for a little bit. Where do you find MHC class 1?
On nucleated cells.
On nucleated cells, everything except red blood cells, good. Where do you find MHC class 2? Thanks, know what that interaction is with different kinds of cells. So like MHC class 1 is going to interact with the T cell receptor in CD4. MHC class 1 is going to interact with the T cell receptor in CD8. Like know those interactions. That's in there. I'll back up some more. MHC class 2 you're going to find on phagocytic cells, right? So what's it going to, that phagocytic cell? It's going to interact with what kind of a T cell? A what T cell? Helper T cell. And then, let's see. Remember I talked about CD4 and CD8? You find CD4 and CD8 where they act kind of like a proofreading. It's not the word I want to use, but essentially make sure the interaction is correct. So just understand that, like, you have MHC class 1, the interaction with the T cell, that T cell is going to have CD8. If you have MHC class 2, the interaction with the T cell is going to be, what did I say, class 2? Class 2 is going to be CD4. That's all I know. Okay. Know where B cells and T cells are made. Know where B cells and T cells mature. Know the different types of antibodies we talked about: IgA, IgD, IgM, IgE, did I say G already? IgD. Make sure you know those, what the characteristics are, how many antigens they bind, where you find them. Yeah. So make sure you know what the characteristics of those different antibodies are. That's literally a whole page of exam. Know the different ways these antibodies work. We talked about a ton of these: agglutination, antibody-dependent cell mediated cytotoxicity, complement fixation, neutralization, opsonization. Know what those are. Those are all in there. We went through those individually, so make sure you understand like what those different mechanisms are through which antibodies act. Just a few questions on that. Know the difference between artificial active immunity, artificial passive immunity, natural active immunity, natural passive immunity. Went through all those. It's on one slide, I think. Know the components of the bacterial cells. So we talked about a bunch of that stuff, like LPS, and what bacteria have LPS, and what bacteria make endospores, and which bacteria have a thin peptidoglycan layer versus a thick peptidoglycan layer, which one has an outer membrane versus the inner membrane, and what flagella are, pili are, fimbriae are. Just know the pieces and parts of the bacterial cell. Know how to do a gram stain. As we talked about it in lectures, I used to try to keep the lab stuff and the lecture stuff separate, but it's kind of hard about. So make sure you know how to do a gram stain. Make sure you know what an acid-fast stain is and how to do an acid-fast stain. The flagellar ranges we talked about, a bunch of those. Amphitrachus, amphilopetrachus, lopetrachus, monotrachus, paratrachus. Make sure you know what those are. That's all. I think it's all in one slide. Know the different oxygen tolerances. Let's walk through the oxygen tolerances. I'm gonna start with the obvious one. What's an obligate aerobe? Yeah, it does. What about an obligate anaerobe? Doesn't, yeah, right, doesn't, can't handle oxygen. Okay, so now it's a little bit harder. What about the facultative anaerobes?
Can't make sounds but not necessarily.
And if it does use oxygen, it does a lot better because it makes more ATP, right? So it's a lot better, it's good. What about microaerophile?
A little bit of
Just a little bit. If it gets too much, it won't survive because those enzymes it makes it deal with oxygen. Free radicals, and they're not that great. Their activity is not real stellar. What about aerotonic? Well, actually, let me go back to microaerophile real fast. So if you grow them in a tube, you would expect them to grow right underneath the surface. Like they're not going to be at the top. That's where all the aerobes are. They're going to go right underneath that, where they can get a little bit less oxygen. What about aerotonic?
They don't care. They don't care for that.
Yeah, they're just going to grow everywhere. They don't care. They can handle it, but they're not going to use it. They typically don't grow as well as the rest because they're not using oxygen. So they're not going to make as much ATP. They don't make as much ATP. They're not going to grow as much. Know those reactions we very briefly talked about for catalase and superoxide dismutase. Ultimately, superoxide dismutase is going to get rid of oxygen-free radicals by converting it to hydrogen peroxide and oxygen. And then once the enzyme gets rid of the hydrogen peroxide, catalase. What's catalase break down hydrogen peroxide into? Oxygen, which is what makes the bubbles. We haven't done this in the lab yet. Everyone in here has done a catalase reaction with Gen Micro, right? Okay. So it breaks down hydrogen peroxide into water. Are you kidding me? Did you guys not do it? That just makes me really angry, to be honest. So some microorganisms make a catalase. So let me ask this to you a different way. No one's in here has had any knowledge? No, someone has. A couple of you had it, right? So where do you find hydrogen peroxide in your body? Like what in your immune system makes hydrogen peroxide to get rid of?
The lysosomes.
Yeah, so you find it in like lysosome, like a macrophage or something. That's one of the ways that some of these phagocytic cells are going to use to actually kill off bacterial cells. Some bacterial cells make a catalase, that way if they end up exposed to that hydrogen peroxide, it breaks down hydrogen peroxide into water and oxygen. So a catalase test, if you grow up a microorganism makes a catalase, and you take a little bit of it off the petri dish and you put it on a glass slide, and you just literally smear a little bit of it onto the glass slide, and you drop hydrogen peroxide onto it. If that organism makes catalase, it's going to take the hydrogen peroxide, break it down into water, which you're not going to really see, but also oxygen. So the oxygen you're going to see is bubbles. So the microorganism makes catalase, it's going to bubble. If it doesn't make catalase, it's going to be really boring. It's not going to do anything. You just drip some hydrogen peroxide on it, it's not going to do anything. Yeah.
Dr. James does those.
Okay. Okay. Yeah, but it seems like there has been some gap in the microbiology curriculum at this institution. Because every once in a while, I'll ask some questions that I'm kind of surprised I don't get answers to. Make sure you know the difference between conjugation, transduction, and transformation. Let's see. Let's talk about those real fast. What's transformation? Natural transformation.
Living in the environment. Environment.
Bacteria picks up.
Yeah, make it DNA.
Yeah, maybe. A lot of times it's fragmented. Okay, good. I don't really talk about this as much. Some pathogens are naturally transformable, which means they can go around and gobble all kinds of make DNA. Some pathogens are not that good at doing it. Those ones aren't. Those ones tend not to be as resistant to antibiotics. More like versus pathogenicity versus virulence. What else? Understanding like legal dose. So what's an LD50? Remember what an LD50 is?
Then there's needed to kill that population.
Right. What about an ID50?
Minimum dose to infect half the population.
Good. Make sure you know the holy crap, I've only done this twice now. Mnemonic. Make sure you know the mnemonic for some killers have pretty nice capsules. Because those are the ones that make capsules, the word ones that make capsules. It's in the bottom of one of the slides. So that will kind of tell you the important ones that make capsules. So make sure you know that. If there was a specific example that we really dwell on in the lectures, like I put a picture up of it, kind of like these things we did towards the end, it's like, okay, these ones block neurotransmitter release. We're talking about like tetanus and botulinum toxin. Make sure you're familiar with the ones that we really have like examples of up there. I'm not telling you to memorize like all the tables and everything like that, but if we talk about like a specific example, like diphtheria toxin blocks protein synthesis. There's a whole big slide where it shows how it blocks protein synthesis. You should be familiar that diphtheria toxin blocks protein synthesis. You need to know all the steps and how it does that? No, but I want you to know that it does, yeah. Make sure you're just kind of familiar with those big examples. Know what the different extracellular enzymes do. Because remember we went down through those, most of them say what they do. A collagenase breaks down collagen, a hyaluronidase breaks down hyaluronic acid. Most of them are also nice red blood cells. Like make sure you kind of know what they do, but most of them are really obvious, like it's in the name of what they do. I literally just gave you an answer to the question. No, I gave an answer to 2 questions. Good job. 3 questions. Okay, awesome. I'm on 50, I mean, that's it. Yeah, that's it. I'm trying to think all this stuff. A lot of this stuff is still review. Like the gram stains, and flagella arrangements, and error tolerances, and LD50s, ID50s. I'm just trying to think, most of the stuff we talk about in Gen Micro, natural versus passive immunities, and interaction between white blood cells and different types of antibodies. Yeah, a lot of this is still kind of reviewed from Gen Micro, so hopefully if you had Gen Micro recently, you should be fine. Is that me telling you not to study? No, study for the thing. But